Hello, histology class. This is Dr. Ogilvy, and I want to give you a few tips in preparation for the midterm by using some of the previous questions. At the outset here, I'm just going to demonstrate some interaction with the virtual slide specimens in Part B, so that if you're failing to do some maneuvers that are very important, that you will not forget to do that. This is one of the actual questions in Part B of Module 1 quiz, where you're asked to identify the tissue within the two regions enclosed by the dashed lines. Well, I do not see two regions here, and the reason I do not is I didn't click on the annotation and then click Go To. That you need to do. So we have indicated two regions, and if you failed to do that, you would have not seen the second region. So now when you examine that specimen, you can zoom up and you can look at both. So first we'll look at this specimen, and this is loose connective tissue. Do you see the very delicate collagen fibers here that are present in the fibroblast nuclei in this specimen? If you go to the other region, then the collagen fibers are even more delicate, and there's lots of space between all of the cells and the delicate fibers. So without question, this is loose connective tissue. Now, in contrast, you had another question. You were asked to name the cell type in this region, but this is dense irregular connective tissue. Do you see the contrast between dense irregular connective tissue and loose connective tissue here? Okay, so that's the first tip or reminder that I would like to share with you. The second is that if you are in this particular looking at a specimen initially, you may not see the annotation, you may not see the macro view, you may see this. And I hope all of you know by now, but if in case you don't, the annotation button displays the question, and the little magnifying glass displays the macro image. So that's a tip. Now I'm going to shift my attention and review some samples of the list of missed questions that I have discovered that a number of students missed in Module 1 Quiz Part A and Part B, and Module 2 Quiz Part A and Part B. And this will be posted along with this video in a bulletin. So I'm going to go through samples of a few of these, and then you'll be able to. The idea is that I went through the module quizzes and I looked at a number of responses in different individuals. And I saw, for example, an answer was lipocyte for adipocyte. Now, we could have given credit for adipocyte. However, you needed to specify whether it was white or brown or unilocular or multilocular. So that's the sort of thing that I'm going to illustrate for you in Part A and also in Part B. In Quiz 1, Part B, there was a question. It was a fill-in-the-blank question where you had to type in the answer. And this is exactly what I was talking about. We only accepted white fat cells, or unilocular adipocytes, or unilocular adipocyte. Just putting lipocyte is not an adequate answer. It is incomplete. And that's why this was marked incorrect. So read the question carefully. It asks for the dominant cell type, and some individuals I noticed put the tissue. They put adipose tissue. Well, of course that's right, but we didn't ask that. We're very strict about this. This course is about details. It's not about generalization and responses. Okay. Another question referring to an image, a static image in Part A of Module 1 quiz. The tissue that occupies most of this specimen is transitional epithelium. And someone put true, and it is simply not true. Transitional epithelium will have many fewer layers of cells here. But most importantly, you're going to see dome-shaped cells, unless we would tell you that the epithelium was very stretched, and then it would be quite thin, only a few layers. This is a classic example of stratified squamous. See, stratified, non-keratinized, stratified squamous epithelium, non-keratinized. There's no keratin at the surface, and you see cells with nuclei right up to the surface. This question reminds me to remind you that knowing the size and the relationship between structures, as was given out in one of the early lectures, I believe on the cell, or it could have been on probably on the cell lecture, in which you're asked to say which of these is smaller or larger than a bacterium. And of course someone answered larger, that a ribosome is larger than a bacteria. That reminds me to encourage you to review the size relationships, and clearly a ribosome is a very tiny, probably about 70 nanometers within a cell, and is not anywhere near a micron or two in diameter, or even a half a micron. So that's very important to understand the size relationships and try to commit those to memory. Now this question, indicating very tiny nuclei that are round and very dense, and the nuclei do belong to lymphocytes. Now perhaps those that missed this question either didn't know that, or know the characteristic features of the nuclei of different cell types, or did not take a close look at this. There are two ways you can take a close look at it. You can actually use the Control plus key on your plus key on your keyboard, and you can magnify it that way and get a closer look, or we reduce that back down, or you can click on the little file name here, which will immediately bring a larger view in a separate tab, and then you can click that back and go to the question. This is another example of a specimen that you probably would benefit from magnifying. By clicking on this, you get a little larger, closer view. You can see the clock face pattern of the plasma cell within that in that nucleus. One last point about this image, which asks the cell type represented by those that are circled and those indicated by the arrow. And those are clearly serous cells. This individual answered erythrocyte, and is totally wrong. But you see the different answers we accepted: serous, serous cell, plural, serous acinar cell, serous demilune, etc. We try to be as fair as you possibly can, and think of all the various answers that could be correct. This also reminds me that on the midterm, you will get a chance to review the midterm, but it will be a narrow period of time, and you'll have to schedule your time to do that. And when you do, make sure, if you think you've typed in the correct answer, you have got to post on the discussion board. This in case will be in the midterm under whatever part, and you need to specify your question number, and you need to specify exactly the phrase of the question and exactly what you typed in, and tell us why you think it may be correct. Okay, here's a question on Part B of Module 1 quiz, and it's referring to a specimen in BioLucid virtual slide, and the answer given here was connective. Well, this is an example of an incomplete answer. Even if this was connective tissue, if you were asked the tissue, you do not have to repeat the term tissue. We try to include tissue in a possible answer just in case someone does. But if you're asked the tissue, all you have to put are the descriptors of the tissue. If it were connective tissue, you would have had to put loose connective or dense, regular connective or dense, irregular connective. Now in this case, it wasn't even connective tissue. It was simple columnar epithelium. And I would like to show you the specimen and point out a couple important things. And the most important one, this is how it would appear when you first clicked to retrieve the specimen. But this is not what we're asking about. And you don't see a space X, and you don't see any other rectangular box. So this should be a very important message to you to click on the annotation and then go to. And when you do that, then you are at where the area and the structures that we're asking for. In this case, what we're asking for is in the box, and it's clearly nuclei very close together. The nuclei are ovoid or oblong, and the cells are taller than they are wide. And so it's definitely simple columnar epithelium. We're only asking about this area. We didn't ask you to go around and look at this, although this is simple columnar, but it's kind of disorganized. If you looked over here, it's still simple columnar, but we chose this one area. So once again, make sure you click on this and go to. Make sure that you also have this macro view so you can see the context in which the question is asked. That's the advantage of using virtual slide specimens on a quiz or exam. And here's a question that refers to your understanding about euchromatin and heterochromatin and what you can predict about the function of a cell. So you're asked to examine the nuclei of two cells indicated by the yellow arrows in this virtual slide specimen and select the true statement about the cells. Well, this is the specimen as you would have seen it when you first clicked on the link. The first thing I'm going to do is click on the annotation and go to make sure I'm actually looking. Actually, it's set at a little lower mag so you can see the surrounding area. This is a sensory ganglion, and these two cells are sensory ganglion cells. They're quite round, and if you increase the magnification up to 100% here, you can see the nucleus in the center here, and you can see the surrounding cytoplasm, and you have a very dense dark spot in each nucleus, and that's the nucleolus. The rest of the nucleus is quite pale, and the chromatin is dispersed, so its euchromatin pattern is the dominant pattern. Now you could enlarge this more by pressing on the Z key on the keyboard and using your marquee, and you can just look at this area at higher magnification. And it's a little blurry because we're looking at empty magnification, but you can still see here that the chromatin pattern is very euchromatic compared to the pattern in the surrounding cells, such as these fibroblast nuclei here, which is all heterochromatic. It's a dominant heterochromatic pattern of chromatin. There's another question about the chromatin pattern in a nucleus. It says, which of the following term best describes a morphological pattern in the nuclei indicated by the arrows in this virtual slide specimen? Here is the specimen. I'm going to make sure that I'm looking at what we wanted you to look at, and we're pointing to three nuclei, and the rest of the cells here, these liver cells, are eosinophilic cytoplasm, and you can see some dark tiny round areas. Those are nucleoli. But the rest, there's a lot of empty space here and light areas, and the DNA is extended. So you have a heterochromatic pattern. So the correct answer to that would be heterochromatic, or excuse me, euchromatic. I misspoke then. It should be euchromatic. And so the dominant pattern in these nucleus is euchromatic, not heterochromatic. Okay, the last question that I want to comment on in Part B of Module 1 quiz is the question that stated two cells at the surface of this are indicated with arrows. They are the same as along the surface of this epithelium. Which of the listed cell types is the correct name for these cells? The correct answer was faucet cell, not transitional epithelium, although the tissue was transitional epithelium. Now let's look at the specimen. You see the same question here, and there are the two cells that are pointed out. And so you needed to, the first observation I will make is that this individual answered transitional epithelium, but notice we asked cell type, not the tissue type. Make certain that you take enough time to carefully read the question. One suggestion I've made to students in the past is just name the subject of the question. Okay, two cells, that's subject, are indicated, verb, with what? Object, arrows. They are the same as along the surfaces. Which of the listed cell types? And you're referring to the cell types here. Just try to dissect the question and take enough time to read it carefully so that you don't miss the points because you misread the question. So we go back to the specimen here. If you needed to enlarge it, this is actually enlarged at 61%. The scale bar here is 100 micrometers. So this epithelium is actually 100 micrometers thick at this point. And we can enlarge this to get a closer look. And if you really need to, you can use your marquee with the Z key and even look at it more closely. So hopefully that will help. Now we're going to go on to part A of Module 2 quiz and part B, and just take a couple on each one of those, and then we'll end this video. Now this question is about the epiphyseal plate, and the statement is the cells in the epiphyseal plate within the yellow circle are active in which of the following ways. So you had to recognize the zones. Over here on the left, the cells are all lined up and they're rather small, and if you needed to look at that closer, then you could do this. But you see these cells are getting larger, and over here you've got cartilage has been deposited to begin to calcify. So going back to the lower magnification, the best answer was cell division has slowed or stopped and the cells are enlarging. But they're not secreting matrix in the form of osteoid. That is occurring over here. This pink material here is osteoid, and it's depositing bone upon the cartilaginous template, that is partially calcified cartilage. Okay, so those are important tips, and hopefully that will be helpful. This question that someone individual missed and answered, that we wanted to have you tell us what these cells on the right, and you can see that this is bone, and if you take a closer look at it, you can see it's bone, and you have the cells lining the surface of the bone here, and here they are at higher magnification, and these are osteoblasts, and the entrapped cells are osteocytes, and you can even see a little extensions here from the cell, and these are canaliculi connecting the osteocytes with one another. Okay, so you have to have remembered, first of all, you have to recognize this as bone. Okay. Secondly, you have to know that those are osteoblasts. Thirdly, you have to know what kind of matrix bone has, and in that matrix of cells and fibers, or excuse me, of fibers and ground substance, what kind of fibers they have. And of course the correct answer is type 1 collagen. I'm going to point out maybe one or two or three questions on part B of Module 2 quiz. And this particular question is asking, it says the cells located between the two dotted lines in this virtual slide specimen are destined to differentiate into which of the following cell types. And the correct answer was chondrocytes, and this individual answered osteoblasts, which tells me that it didn't recognize that they were looking at cartilage and not looking at bone. So let's look at the specimen. This is the specimen, and I'm going to make sure that we're looking at the area we wanted you to look at. And this is what it is. So you have to recognize that this is cartilage, and if you needed to magnify it, you have plenty magnification here, and you can even enlarge it with the pressing on the Z key and making a marquee. And that's way over magnified. So you look on the edge of cartilage, and if you remember from lecture and the lab that cartilage, except for articular cartilage, has a perichondrium tissue that is covering the surface. So the cells located between the two dotted blue lines differentiate into which? We don't even want to name the cells. It would be chondroblasts would be the ones that are looking more plump, and over here, back here, there would be the stem cells. So they're going to get larger and larger, and they're going to destined to become chondroblasts. So the best answer is chondroblasts. Or excuse me, chondrocytes, to differentiate from chondroblasts in the perichondrium into chondrocytes. Excuse me. Now, if you notice here, we're actually on the edge of a structure that has a very large lumen. This happens to be the trachea, and this is the esophagus. And if we back down at low magnification, you can see that over here. Trachea with hyaline cartilage all around it, and the esophagus. And also lining that trachea is pseudostratified, ciliated, with the cilia projecting columnar epithelium. If I enlarge that slightly, you'll see that. And we go back down to lower magnification, and here is the esophagus. And it is lined with stratified squamous, non-keratinized epithelium. You see the cells go all the way to the surface, and they're flattened, but there's no keratin here. Stratified squamous, non-keratinized epithelium. The last question that I'm going to use as an example of one that's missed and why it may have been missed is this question. Question 11. Several examples of a structure are encircled. Select a correct identification. The correct identification was center of osteons. Now you had choices in this multiple choice. I'm not showing you all the choices, but the correct choice was this. There was no choice that said Haversian canal. So you had to translate by, first of all, looking at the specimen, making sure you're in the right area, like that. So we have these structures encircled, and we look at them and see, for example, this one. This has lamella concentrically around it. So does this one, and this one over here. So those are Haversian canals. And the question asks, select the correct identification, and the correct choice was the center of osteons. The Haversian canals lie at the center of osteons. You could have also looked around and looked at more examples, because we just pointed out a few, but there are many, many examples of Haversian canals at the center of osteons in the specimen. And over here, this is all bone marrow. If we go down to lower magnification, you see we've got a mixture of adipose and erythropoietic tissue in here, where some red blood cells and white blood cells are being produced, but it's very dominant with adipose tissue. Okay, so that includes, hopefully, you will have some assistance in the way to approach these unknowns, as well as other questions as you prepare for the midterm exam.